So I'm going to set this to record now. All right. Let's go ahead and get started today. So it's my great pleasure to introduce our speaker today, Dr. Loreen Stromberg. It's typically when you're going to introduce a speaker, and I caught myself like, oh, I got to go and sit down and write something. I was like, well, I don't have to write anything down. I've known Loreen since grad school. We've been working together for so many years now. Like, I don't have to write anything down. I just got to introduce my friend and colleague here. So I met Loreen when she was in grad school at UNM and was working on a project co-sponsored with the labs with a colleague of ours that she's working with again now as a prestigious postdoc fellow I'll talk about. But she was a graduate student in bioengineering then, having already finished her degree in biochemistry, which and I bring that up because it really describes part of this path that she's been on through her career, one that I can very much resonate with, but also I think is why she does such amazing work in science and engineering, because she has a very broad background and base of understanding. And during I'll brag about her because when she was defending her PhD thesis, it was one of the best I've ever heard, and she graduated with distinction, and that is something that I do not look on lightly. And at UNM was a very rare occurrence that we actually would graduate somebody with distinction in their PhD thesis. But it was tremendous, and we all agreed that it was just absolutely tremendous work. And she went on from there to Iowa State for a postdoc, where again she continued this path into exploring new branches of science. I think in mechanical engineering, so making this transition again, biochemistry, bioengineering, mechanical engineering. And while there, actually was the CEO and founder of a startup company, and NanoSpy, and did a bunch of patents and publications. Just had a tremendous time out at Iowa State before coming back to Los Alamos as a Reines postdoc fellow. And so these are really, really prestigious. So Lionel has, I think, maybe 4 of these that are individually named, but only one of these every year is awarded. And it is, so these are the best of the best of the best. These are just tremendous honors when you receive a distinguished postdoctoral fellowship. I wouldn't have even been brought up for consideration even talking about one of these things. These things are hardcore, and Loreen is incredibly deserving of this honor and award, and has done tremendous work there at Los Alamos that she's going to be telling us about today. I'm not going to tell your story, although I loved it, Loreen, in your bio with Thor's hammer, because I thought that might come up sometime today. But if not, I encourage everybody to go read her bio because it's really funny. She has a funny anecdote there about representing Los Alamos at a DOE competition. But anyway, so it's my great pleasure to introduce Loreen. She's going to talk to us today about interfacing biomarkers and materials science, applications for detection, therapeutics. So thanks for being here, Loreen, and it's all yours.
Thank you for that very generous introduction. I hope I do it justice today. And thank you for the opportunity to speak today. I put together this talk with the theme of actually highlighting how material science and chemistry affects the field of assay development. And if we ignore those interactions, it can actually cause us to misinterpret our results if we don't fully understand what is going on. So I've actually had the opportunity to work on a lot of different projects, which Gabe kind of highlighted in the introduction. And so they've been funded by a variety of organizations, and you can see all these different fields that I've worked in. And so when I came to designing this talk, I had to pick some of my favorite examples or good examples, because the majority of my work actually links back to assay development. And my view of biomarkers and microorganisms really plays a role, or in that interaction of, I guess, material science really plays a large role in how I approach these projects. So I hope I've picked some interesting examples for you today. So starting out, when I think about developing an assay for biological organisms or biomarkers, I have something similar to this wheel actually in my head. First, you have to know, it's ideal if you know what kind of pathogen you're looking for. Is it a virus, a bacteria, a fungus? Does it have any unique markers that are associated with it that you can readily use for detection assays? And what type of molecules are those? Are they proteins, lipids, sugars? And how do those present? Is there a unique presentation during the timeline of infection that you can basically naturally exploit to create a biomimetical functional surface for detecting those? It's also equally important that you have access to ligands that can specifically bind your biomarker of interest, because you're going to need to characterize those interactions in different ways. Otherwise, if you don't have access to these ligands, you might end up going down the route of having to make your own or having them made, and that can be very costly and time consuming. Depending on your assay platform, you may need access to stable reporters, such as fluorophores or quantum dots, so that you can get that feedback in your signal to noise ratio. And of course, you need to validate your assay in the target matrix. So whether that is a food or a clinical sample, you need to make sure that you're getting the appropriate signal to noise ratios that are going to meet the necessary threshold of sensitivity and specificity. So while I've actually pictured it here kind of last in the assay, thinking about that target matrix and how that biomarker is going to interact with it comes along very early in the thought process for designing these assays. So once you've done all that and you've benchmarked your assay and validated its performance in your target matrix, if you're interested in commercializing it, you need to go through this somewhat costly and time-consuming process of external validation. And I've listed AOAC here. It's a common company that does external validation for foodborne pathogens, which I've worked a lot with. So as I said, I worked a lot with foodborne pathogens. And one of the really basic reasons that my research has focused on that is there's actually a lot of money available to do research in this field. And that's because our food supply is inextricably linked to our national security. Last time I checked, there were about 227 known diseases that are linked to bacteria alone in our food supply. And there are not really any food groups that are going to be immune to this. Basically, it's going to impact you whether you consider yourself a carnivore or a vegan. And because all of these different food groups have very different compositions, if you develop an assay for produce, it probably isn't going to work in meat. And this is a really fundamental problem for foodborne pathogen detection. There is no universal approach that's going to work for every matrix. So obviously, different pathogens have different biological signatures associated with them, which is often based on their basic structure. A lot of my own personal work has focused on gram-negative bacteria, which, as you can see, have a really distinct structure as compared to gram-positive. This outer membrane layer is full of unique markers that make it distinct. So it's really straightforward to be able to use these for detection. So my first case example is going to be looking at the biomarker lipopolysaccharide, interfacing that with lipid bilayers to facilitate detection. Our first example looks at Shiga toxin-producing E. coli in beef products. So you may hear me refer to this as STEC, which is habitual because I did a lot of this work in graduate school. And after referring to it as STEC for 4 years, you can't break that habit. So the problem associated with STEC detection is that in 2012, the USDA actually started to require screening for 7 different serotypes of STEC in beef products. And a really common example of a serotype is E. coli 0157H7. I'm just introducing you to that terminology and getting you familiar with it because in a few minutes, I'll be talking about different types of lipopolysaccharides and their O serotypes. And I want you to understand where that term comes from. So the problem with STEC detection is that there were no standardized detection methods, no standardized sampling methods for how we were going to collect these. And there were no commercially available antigens or antibodies, but we still had to develop assays for these that were able to discriminate between these different serotypes. So 2 of the predominant biomarkers that we chose to look at was lipopolysaccharides, or LPS, and Shiga toxins. So LPS covers about 75% of the outer membrane of gram-negative bacteria. And because it is so predominant, it's actually often released during cell replication or lysis. It's made of 3 distinct components. The O antigen here is the part that's variable and different between different serotypes. So that's what facilitates discriminative detection. And then there's also the core polysaccharide and the lipid A group. And these are fairly well conserved within a species. Lipid A is also known as endotoxin because it's highly antigenic and it's responsible for causing sepsis in cases of like septic shock. Because of this amphiphilic biochemistry, LPS is also known to associate with lipid carrier molecules in vivo. So it has its own LPS binding protein. It's also going to associate with your cholesterol lipoproteins like HDL and LDL. But when these molecules aren't around in an aqueous buffer system, LPS actually presents as a micelle. And the size and shape of that micelle is highly dependent on the environmental conditions that it's presented in. So our problem is we need to be able to detect this O antigen. So we have to present the molecule in a way where our antibodies or our ligands can bind this and discriminate between different types of LPS from these different stacked serotypes. So the platform that we chose to use is called the waveguide biosensor, which was developed at Los Alamos National Lab. And this platform uses a single mode optical waveguide. And in case some of you may not be familiar with what a waveguide is, it's basically a piece of very thin piece of material. It has a very high refractive index, and it's surrounded by material of lower refractive index, in this case, glass. And then we can couple light in through a grating, and that is going to result in total internal reflection of the light down the length of the waveguide. No system is 100% perfect. Some of that light is going to escape in what is called the evanescent field. And this is about 200 nanometers in distance away from the surface of the waveguide. And right at the surface where this field originates allows us to perform like very sensitive biodetection. So in the case of LPS, what we do is we functionalize the surface of that waveguide with a lipid bilayer. It allows for insertion of amphiphilic molecules such as LPS, and we can come along and detect that with a fluorescently labeled antibody, thereby facilitating discriminative detection between these different LPS types. So here are some of the data from that platform for LPS detection. On the left, we are showing a benchmark assay using LPS from 0111B4 serotype. And we're detecting that with a polyclonal antibody that's labeled with Alexaflor 647. On the y-axis, we have the signal and relative fluorescence intensity that's reported in the thousands. And on the x-axis is the wavelength that we're recording each one of those signals at. The NSB is the non-specific binding. It's basically the noise of this assay. So that's any non-target or non-specific interaction of the antibody binding to the lipid bilayer. When we perform the calculations, we can determine that the signal to noise ratio of this assay is about 124. For reference, normally we would consider a signal to noise ratio of 2 to 1 to be positive on this platform. So this assay is working much better than we really planned. Of course, after developing all the benchmarks and all the methods for handling LPS and putting down the lipids, we of course need to test the performance of this assay in our target matrix, which in this case is ground beef. So that's over here on the right. We're detecting LPS now from 0157 in a ground beef homogenate. And to perform this assay, we froze ground beef and ground it up even into a really fine powder, essentially, and then suspended that in buffer. And we can spike LPS into it. And we found that we can pretty reliably perform this assay with a 1 mg per mil suspension of ground beef. And you'll notice here that the intensity of our signal is actually much lower, indicating that the antibody used in this assay didn't actually perform as equally well as the benchmark. However, if you examine the signal to noise ratios of the benchmark to the beef lysate, you'll see that in 2 cases here, that signal to noise in the beef lysate is actually better than the benchmark. So we had to do several experiments to investigate this. And what we came up with is that the beef lysates actually provide an additional blocking step against that membrane to reduce any non-target binding with that antibody. And also, this is a more relevant matrix. So it's very possible that LPS is associating with proteins in that matrix that facilitate a more physiological presentation and binding by that polyclonal antibody. This data here demonstrates that we can actually use this method to facilitate discriminative detection of LPS using serotype-specific antibodies that were developed in the course of this project. So remember for a second here that this O antigen portion of the LPS is distinct between serogroups. So these antibodies are binding these different sugar portions here on the O antigen. In the case here of detection of LPS 0104, we're using a polyclonal antibody, and that has many more paratopes as compared to that of a monoclonal antibody, which is being used to detect the LPS 0111. So that's why we're getting a much higher signal with our 0104 as compared to the 0111, because there's that antibody can just bind many more epitopes along this region of the LPS, which is going to facilitate that order of magnitude sensitivity in our limit of detection. So I've spent, and this, of course, this platform doesn't just work for detection of amphiphiles. We can also use it to detect proteins. So in this case, Shiga toxin. So in order to do this, we can functionalize that lipid bilayer using some different strategies. So for instance, Shiga toxin has an A subunit and a pentameric B subunit. And that binds the global triacylceramide or GB3 receptor on a cell membrane. So we had kind of two immediate options to functionalize the surface. We could either insert GB3 into our lipid bilayer, and then that would also facilitate multiplex detection because we could detect LPS at the same time that we're detecting Shiga toxin. But unfortunately, GB3 is actually really expensive. And so it would drive up the cost of our assays significantly. And also, we didn't quite have any information about how LPS might interact with bilayers that are enriched with GB3. So we actually chose like a more expedited, simplified method of putting a biotinylated head group in our lipid bilayer, which is pictured up here in the right. And what we did is you create a capture site by putting down streptavidin, which binds that biotinylated head group. And then you can use a biotinylated antibody to create a capture site for your Shiga toxin, and then come back and detect that with a fluorescently labeled antibody. And so that's what that data down here in the lower right is. Basically, this is how we're approaching detection of Shiga toxin 2 in this scenario. And when we calculated out, the signal to noise ratio is 16 for this benchmark assay. And we still use this detection motif for a variety of protein antigens in our lab. So I've spent 3 slides telling you about the results of this assay, but really I want to talk more about the functional surfaces and how these biomarkers are interacting with surfaces. So before I started this project with LPS, the lab group had really laid the groundwork and performed assays with a similar biomarker from tuberculosis called liporhabdomannan, or LAM. And when they did the characterization part of these assays, it was found that LAM partitioned into bilayers quickly, and that was recorded with fluorescence microscopy and atomic force microscopy. And this up here in the right is just a schematic of how those experiments are performed. You have a clean glass cover slip, you put down a sample barrier to create a well, and then you can exchange various amphiphiles or proteins or lipids in and out of that and use an inverted imaging system to document your interactions. However, when Gabe and his postdoc went to do the same experiments with LPS, we discovered that LPS actually punches holes in these lipid bilayers. And I always have to put up a picture of an AFM figure because no one ever believes me that these are holes, but they are. We've characterized them extremely well. And in our heads, what it has immediately created the question that perhaps this mechanism is actually occurring inside of our waveguide flow cell when we're performing our LPS assays. And that might be the reason why we're getting such a high signal to noise ratio. So what we ended up doing was creating an analogous system to the flow cell that we could image through. And we discovered that actually below 50 micrograms per mil of LPS, which is the more relevant concentration, none of these LPS serogroups actually caused any hole formation inside of the flow cell. In fact, the only one that did was the commercial LPS 0111B4 that we were using for all of our benchmark studies. So that was good news. We didn't have to throw out a bunch of data, but it really helped us understand our system. And it provided some really cool ideas for ways to pattern membranes and perhaps improve our assays. And this mechanism of hole formation has always been in my brain, because the more you study it, the more interesting it becomes. So for instance, in the process of these experiments, we discovered that LPS-induced holes are pretty persistent. And what I mean by that is that you can backfill these a number of times with lipid and re-incubate with LPS. And in general, the holes will recur in the same positions. And that's what this iterative image analysis over here on the right says. Basically, the holes do not always recur, but when they do, it's always in a position where there was an initial hole. No new holes are formed. So then we started to exploit this by pairing it with microcontact printing techniques. So here we have substrates that are pre-patterned with a grid, and then you can deposit monolayers and bilayers here. And basically these are the same things, just a different shape. And LPS is going to interact at that interface of the grid to create these little triangular or diamond-shaped holes. You can refill these with different lipids or different proteins, which is pretty cool to be able to pattern a membrane like that, because getting things close together in a membrane is going to allow you to study real, more biomimetic interactions that happen at a cell surface. But this question of hole persistence and hole formation has always nagged me. And so what I'm really working on right now is different methods to try to identify the actual mechanism of this hole formation and trying to synthesize it and inkjet print it into a variety of patterns. So we look at a variety of lipids and a variety of LPS serogroups and pattern them with an inkjet printer, and then study them using the same open air system. And also, we want to be able to basically increase the throughput at how we study these systems. And so we recently acquired a very fancy plate reader with automated imaging called a Citation 7. And so we're able to put these lipids into glass bottom microwell plates and basically screen a whole bunch of different compositions to analyze whether we're seeing this interaction or not. And this informs not only my work, but work with the waveguide biosensor for various assays, and then other work that team members are doing developing therapeutics for sepsis. One of the problems with this is that the automated imaging on the Citation 7 really likes to focus on bright spots as opposed to the dark spots that we might be interested in. So we're working with a company to try to kind of tweak their algorithms a little bit to see if we can make that work for us for a better throughput screening method. We're also putting these compositions onto fluorescently doped microspheres, and we use flow cytometry to evaluate whether there's any dye leaking out of these microspheres after we incubate with various amphiphiles. This is also going to be a more clinically relevant application for these membrane insertion type assays, because a lot of clinics use flow cytometry as their platform. So there's a lot of advantages associated with membrane insertion. Basically, it facilitates that biomimetic presentation of LPS, and it allows us to discriminate between different O antigens, whereas that's not really a thing in the current marketplace right now. You can do various testing with intact bacteria, but telling the difference between two biomarkers with a detection assay is not really available. However, the sensitivity of this assay doesn't really meet the requisite for industry. So that's about 10 colony forming units or CFU per mil of bacteria. And there's 62 picograms of LPS per cell. So if you do the math, our limit of detection isn't where it should be to meet this requisite. The incubation period is also very long for LPS. It's a big molecule, so it can take up to 2 hours to associate with those bilayers and get the necessary signal that we need. But we think that pre-patterning the bilayers is actually going to improve the speed of LPS association. Another drawback is that these bilayers can have a short shelf life. So we have other people in our group that are looking to create air-stable bilayers. And of course, tuning our lipid compositions, not, like I said, doesn't just inform my project, it informs a lot of other projects currently going on in the lab. So the next example is actually one of my favorites. Basically looking at different materials and how to combine those to facilitate detection of salmonella using something called inkjet printed graphene. So salmonella is a big problem, and it's pervasive in the food chain. It causes, without fail, at least one major outbreak a year. Usually it's more like an average of one a month. And one of the problems with salmonella detection is that it just takes too long to detect it, or the current methods are not sensitive enough. So this is a generalized schematic for how food companies test their samples. So in the case of a solid sample, you would swab that or homogenize the meat. If it's a liquid sample, you collect an aliquot, and then that's put into a buffer to stabilize and preserve it until it gets to the laboratory. So then after that point, it is basically inoculated into media and grown overnight to amplify the number of bacteria. And then it's plated onto a selective media, which is only going to allow for detection or for growth of salmonella. And then after you incubate that again overnight, you pick off your colonies and you are going to use another confirmatory test like PCR to identify them. And this is required because the test just cannot directly detect the low 5 CFU per mil of bacteria without amplification. So when we think about developing a detection assay for salmonella, it's a little bit different than E. coli in a couple aspects. There are 3 primary serovirus of salmonella that we're mostly concerned with, and that's typhimurium, enteritidis, and typhi. That last one really isn't of much concern in the United States, but it is in other developing countries. So the key difference between E. coli and salmonella detection is that there are beneficial types of E. coli. Like some of them are just good for us, some of them have varying virulence. And so it's important to be able to discriminate between them. But there are no good types of salmonella. There's only positive versus negative and live versus dead. So while salmonella are gram-negative and they actually have LPS, just like STEC, in this case, we're interested in detecting whole intact bacteria. So our previous platform wouldn't really work for that. So in this case, we want to use an electrochemical biosensor and attempt a label-free detection method. And as Gabe indicated, I worked with this group at Iowa State University, and we were developing this method for inkjet printing of graphene to make electrochemical sensors and printed electrodes. And basically, the standard method for any conductive ink is that you would bake it at a very high temperature to cure it. But what we want to do is laser treat it, to anneal it is what it's called, or to cure it. And we found out that when we laser annealed these printed electrodes, it resulted in the evolution of these pyramid-shaped graphene structures on the surface of the electrodes. Additionally, by adjusting the energy density, what's called laser fluence of the laser, we could actually tune these structures to induce superhydrophobicity. And when you examine like the corresponding sheet resistance of these different laser treatments, you discover that the highest laser energy density results in the lowest sheet resistance, which is really good for improving the sensitivity of your electrode. Additionally, these materials demonstrated excellent sensing capabilities for peroxide, as you can see down here. We're detecting very low micromolar amounts of peroxide that are spiked into about 10 milliliters of buffer. And they have a great linear dynamic range for sensing. The problem with this is that this format of electrode really kind of tracks the movement of electrons as they move on the surface. And we want to detect salmonella. So we wouldn't really expect any net change in electrons when we're doing our detection. So we needed a different kind of electrode. And we selected an interdigitated electrode, or IDE, which is pictured right up here on the left. And what we wanted to do was print these IDEs and then functionalize them with capture ligands and perform detection of salmonella. So that you understand how this works, basically an IDE is 2 separate electrodes, and they have fingers that are intermeshed, but they don't touch each other. If they touch each other, it actually causes the circuit to short and your electrode will fail. Additionally, the closer together these fingers are, the more intense that electrical field is where you're going to be performing detection. So we need a really high resolution IDE, but inkjet printing conductive inks like graphene is known to have some problems, such as this like wicking coffee ring effect, or what's called satellite droplets, which is what happens when your ink hits the substrate and then spatters. So any one of these things can cause our circuits to short. So we need a new method for printing graphene ink that has the resolution we need to achieve lots of those IDE fingers that are close together, but not touching. And what we come up with, we coin inkjet maskless lithography, or IML for short. And how this works is instead of printing the graphene ink directly, we print a reverse pattern in a really nice jettable polymer solution. You allow that to dry and then spin coat a layer of specialized graphene ink on top of it. The substrate is baked at a low temperature to burn off any solvents, and then you lift off the polymer and your pattern is revealed underneath. So here is a video of that. Basically, you dip the substrate in an acetone bath, and then you can use an acetone spray to remove any of that underlying polymer, and your pattern is revealed underneath at a pretty good resolution, actually. So then you allow that to dry and the acetone to evaporate off, and then you can proceed with laser annealing and functionalization. So originally, we actually wanted to try to use aptamers for detection, but no matter what I tried, I could not get that to work. So we actually settled on an anti-Salmonella antibody. And as you can see down here in the lower left, this printing process actually produces a large number of IDEs in one batch. The fingers of these IDEs have the resolution necessary to facilitate sensing. In this case, each finger is about 75 microns wide. They're 50 microns apart. However, when we tried to anneal these with our laser technique, the laser was actually just too powerful, and it would cause the graphene to just blast off the surface because these fingers are very delicate. So we developed a technique using a much smaller, less powerful laser to anneal the graphene. And as you can see here in E and F, we're no longer seeing those pyramid structures that we really wanted to work with. Instead, our graphene actually appears spongy instead. But we believe it still has adequate surface area to functionalize. But of course, we need to characterize it to make sure that we can attach or covalently attach antibodies. So we sent off some samples to do X-ray photoelectron spectroscopy to analyze these surfaces. And what you can see here in the pre versus the post laser treatment is that basically the intensity of these sp2 carbons has decreased and the sp3 alkene carbons have increased. We also see the evolution of these groups that we can identify as carbonyl and ether groups, which are most likely occurring right at the surface and edge planes of the graphene structures, which is right where we want them to be able to functionalize these surfaces. So once we had some working IDEs that were performing well in standard tests, we wanted to compare their performance against IDEs made of other materials. So we looked at microfabricated platinum IDEs and an aerosol printed graphene IDE made by one of our collaborators. If you're not familiar with this type of data, these are called Nyquist plots, and they are generated from something called electrochemical impedance spectroscopy. And the benefit of this type of data is that it can show us very small changes in the surface of our IDEs. So that's what's facilitating label-free detection. On the x-axis, we're looking at what is called real impedance versus the y-axis, which is what's called reactance or imaginary impedance. And for the purpose of sensing in this application, what we want to see is that as the Salmonella concentration increases, the diameter or change in real impedance should be getting bigger, because that indicates that our charge transfer resistance is increasing. And in the case of all these materials, that's exactly what we see, with the exception that we can't quite tell the difference between 12 and 16 CFU per mil of bacteria. But also, we can't really confirm exactly how many bacteria are binding to our electrodes. So we were not too bothered by that. we were really pleased to see, though, there was a very large difference between the antibody and 5 CFU per mil, as well as 5 and 12 CFU per mil, indicating that we can perform this sensing very reliably, even though we are doing detection in 100% chicken broth spiked with bacteria. So the question is, can these other electrodes also sense salmonella at those low levels? It's very, very possible, maybe even likely. However, we're trying to produce sensors that have a good economy of scale. So these super high quality microfabricated platinum IDEs are really pricey to make. They have to be cleaned with piranha before you use them. You have to put down like a self-assembled monolayer to attach the antibody. So there's a lot of steps for potential error in using these. And also they're very expensive. These aerosol printed electrodes created by our collaborator, they use their own proprietary ink solution. But that ink had to be thermally treated in an oven at a minimum of 400 degrees centigrade. So that really limited what kind of substrate that we could use. So basically, we're limited to things like silicon wafers that can withstand that temperature. And so for reference, our own IDEs on printed or IML printed graphene IDEs are actually made on a chemically treated plastic. And in total, that costs a little less than a penny to produce. So when we originally started to collect signals from our electrodes, we saw what we interpreted to be like these imperfect semicircles that we thought was a lack of stability in our signal. But when we really dug into that data and teased out what was going on, we figured out that the sensitivity of these tests is actually just detecting the heterogeneous surface of our electrodes. It's facilitating that sensitive detection. So our IDE designs had a lot of advantages. Of course, we were able to meet the industry standards, and which is why this technology was taken by a company and adapted and in development for marketing. But we couldn't get a recognizable signal when using aptamers, so that was a way to potentially reduce any batch-to-batch error in our detection signals. Aptamers are slightly cheaper than antibodies as well. So that was a bit of a drawback. Also, our material is very strong. Graphene is very strong and conductive. So it holds up very well in complex matrices like the chicken broth. A drawback is that the more hydrophobic your surface gets, the more difficult it is to functionalize. Because when you are trying to put an aqueous solution with your antibody on that surface, it will literally jump off the super hydrophobic surface. So laser annealing versus our thermal annealing allowed us to use a variety of substrates, but our original process was too powerful. So while we didn't get those great pyramid structures, we instead got spongy graphene, and that still worked for us. We also obtained a patent very, very quickly for that new printing method for conductive inks. The full patent was issued in December. And that's because our original inkjet printing process was too low in resolution. So it really made us innovate a new way to print. And of course, our sensors were cheap, which makes them marketable and scalable. So our very last example that I'm going to talk about today is actually a little bit different as far as not detecting foodborne pathogens. We're going to talk about actually creating a functional surface and ligands to evaluate bacterial viability. And that's because a lot of my very recent work at LANL has focused on developing a new kind of therapy to combat antimicrobial resistance. And unless you are living off the grid, which you're not because you're watching me right now, I am sure that you are aware that antimicrobial resistance is a huge problem. So I'm not going to talk a lot about that because just to give you basically this estimate, if we don't come up with any successful methods to curb antibiotic resistance or no new drugs to combat it, which is actually very likely, then by 2050, we should be experiencing approximately 10 million deaths per year that are directly related to antimicrobial resistance. And when you look up the current economic costs of this, it's devastating. So we really, not only for the people factor, but also for the global economy, need to come up with ways to treat this. So a team at LANL put together this idea that we want to try to use medical radioisotopes to develop a therapy for this. And the way we want to do this is to use what are called alpha emitting radioisotopes, and these are free metals. Basically, an alpha emitter releases what are called alpha particles during their natural radioactive decay process. These particles are capable of delivering a very high energy dose in a very localized manner. And this has actually seen some really successful results in trials for treating cancer. So the way that has been done with cancer is to basically use a medically approved chelating agent. And then you attach that to a targeting ligand, which is usually an antibody. You then chelate your radioisotope and deliver it. However, we're trying to use a method that's tailored to targeting bacteria, not cancer. So our approach is to use siderophores, which are small molecules created by bacteria. They go out and scavenge free iron, and they bring it back to be internalized by the bacterial cell for metabolic purposes. So we purchased 3 different siderophores: enterobactin, pyoverdine, and desferrioxamine. And we use them to chelate the alpha emitter known as thorium-227. So what is our problem with this? Basically, our problem is when you do studies to evaluate antimicrobials, you have to evaluate bacterial viability when you're done. But how do you evaluate the bacterial viability of a solution that's very radioactive? So just to give you a quick view of basically our growth curves, what we did is we looked at Pseudomonas aeruginosa, and we tested 2 different doses of 0.5 and 0.16 microcuries of thorium. And we looked at 6 different concentrations of each of those siderophores, and we tracked the growth of a strain of Pseudomonas over 24 hours. So reported here on the y-axis is the optical density at 600 nanometers, which is recorded every 10 minutes over that 24-hour period. And in each graph, we have a control of the untreated bacteria, which shows up as the blue dotted line. So in graphs B and C, we're actually comparing that natural thorium-232, sorry, as well as iron to the behavior of thorium-227. So there's a lot of graphs associated with this, so I've only presented a few of them here, and we don't have a lot of time to discuss them. But I'll just summarize for you that what we see actually in nearly every single plot is that there's a significant bacteriostatic effect, which we do not see in just the siderophore controls. However, like I said, we wanted to evaluate viability. And when everything is super hot or radioactive, that's a lot easier said than done. So one of the first methods to attempt to do this is basically I spent a long time processing this data to see what it could tell me in a quantitative manner. And I came up with the fact that if we can extract the change in growth rates over time, that really gives us like these little dynamic snapshots of what the bacteria are doing at any given point in those growth curves. We can also precisely identify the lag phases of each condition to facilitate side-by-side comparison. And so sure, this data actually gives us a really good snapshot of viability, but it doesn't give us those solid numbers that microbiologists and big pharma like to see. So really, the standard methods for doing a viability assessment would be, of course, the gold standard is to basically plate out your bacteria after you're done with your experiment and count how many you have left. And we actually did quite a bit of this, but the problem we had was that we had to minimize the amount of time that we're handling these cultures. We couldn't perform 6 to 9 dilutions of 96 wells for each replicate and then plate them out. So we had to estimate, and sometimes we estimated wrong. So there were too many to count, or they were completely absent. And we also tried using ATP luminescence assays, and our standard curves were great. But when we compared these to the growth curves, it would lead to these wildly different values because there's different ATP contents at various points in the cell's life cycle. So that left us with the bacterial viability assay, which is a live dead stain. And so in general, this would not be a quantitative assay because you would use microscopy to evaluate it. And I really wanted to use flow cytometry, but we don't quite have the capabilities yet of pumping radioactive cells through the flow cytometer. So, and the plate reader, which seems like a really intuitive and obvious solution, actually there was a lot of inherent problems when we tried to evaluate the fluorescence of these live dead cells, because Pseudomonas actually pumps out a lot of fluorescent compounds while it's growing. So that would interfere with our results. So back to microscopy. Our solution was to try this method in a paper that was previously published by a group here at LANL. And a basic overview of that method is that we wanted to form a pattern self-assembled monolayer on top of a glass slide, and basically using amine protected pegs and methoxy terminated pegs, which allows us to control the percentage of surface functionalization. And because this is in a pattern, you end up with a very defined area that you can quantify. You perform a series of chemical reactions with the goal of covalently attaching biotin to the surface. So here in this first figure, you see AFM figure, you can see our self-assembled monolayer, or SAM, with 2% biotin. And then the next, we have streptavidin bound to that biotin that we're detecting. In the paper, they were actually able to biotinylate siderophores and attach them to the surface. And then they compared that to the antibody. Because only viable bacteria should bind to a siderophore, we thought we'd be able to use this to quantify viability fairly quickly. And the antibody binding would determine non-viability. So by teaming up with the space science group, the people who wrote this paper were actually able to do some image analysis and quantitate the data that was generated, which is what you see down here in the bottom. However, when I attempted to recreate this technique, I was able to pattern the surfaces just fine. But when it came to biotinylating siderophores, it ended up being extremely difficult, and we couldn't repeat the method. We even talked to the original author of the paper. But no matter what we did, myself and the chemist couldn't confirm if biotin was actually cleanly attaching to the siderophores. We ended up with some very convoluted mass spec data as well. And so we spent quite a bit of time on this, which was last year. So we kind of ran out of time. So we ultimately decided not to pursue it because we might still be left with the issue of trying to incubate a radioactive solution on top of a microscope slide and then imaging it in a way that didn't contaminate everything. So we were back to basically just the live dead stain and some pretty pictures. But in the process of trying to drive this forward and innovate, we came up with a fluorescent siderophore conjugate that we could use to label bacteria and visualize them. And that's tested around, so that's the images here on the left is a desferrioxamine siderophore conjugate with FITC. And as you can tell, especially when you look at these confocal stacks, which I can't seem to pause here, sorry. Basically, there's definitely brighter parts of the bacteria where we would hypothesize that these siderophores are localized to. So ultimately, though, we have a pretty picture of live and dead Pseudomonas. So the dead is the red bacteria, the green is the live. And as you can see, there's a lot of really happy bacteria here, which is actually okay because this is our lowest dose of thorium and the lowest dose of siderophore. And if you're really interested in this project because it's fun and cool, we will be putting out that paper very soon in its entirety for you to read. So this technique that we use really should have been able to detect viable bacteria via siderophore binding, but we couldn't replicate that conjugation of the siderophore with biotin and get that to incubate on the surface. And we would have been able to provide a very quantitative method compared to the lengthy dilutions and overnight growth of culture methods. Also, it would have minimized waste, which is important in a radioactive space. And ultimately, we're not sure if we could have come up with a system that would have been compatible for using with radioisotopes, but honestly, if we had something working, I'm pretty confident we would have. But again, troubleshooting that technique caused us to come up with something new and innovative, which is that fluorescent siderophore conjugate to label bacteria. And we definitely have some plans for using that in future experiments. And the lack of easily accessible methods for determining viability caused us to perform this in-depth data analysis, which we're going to continue using whenever we do bacterial studies in our lab. So kind of some overarching thoughts and my perspective on things is I've encountered a lot of trouble in my research career thus far, but I actually don't look at it like trouble anymore, because it's forced the adaptation of so many new and cool techniques. So if I'd given up, for example, on our LPS assays because of incubation times, we never would have discovered what it does with lipid bilayers. We never would have discovered patterning methods for it. And if we'd given up right away on printing conductive graphene inks and just used a commercially available electrode, we wouldn't have, innovated and come up with a new printing method that was rapidly patented. So again, why is that interaction with the surface so critical with biomarkers? So like I said before, we thought that our original Nyquist plots were actually not stable. But when we evaluated that data and did experiments, what we discovered is that what we perceived as instability in our signal was actually just the assay performing as it should in evaluating that surface area of our electrodes. We're really working to tune the surface composition of our different lipid bilayers for various ampliphiles in our waveguide assays, as well as for membrane patterning. And so one of the reasons we want to do that is because if there's other holes that are formed by various ampliphiles, we want to be able to prevent that. And because that's a more biomimetic system. And that's just going to feed both our information for assay development, as well as other groups and, other people on our team in their effort for preventing sepsis. So I presented approximately a decade worth of research here. There has been a lot of different people that have contributed to these projects at various points in time. I got to, of course, thank the LANL LDRD-DR program for their radioisotope alpha emitter therapies, and the named the named fellowship postdoc program for supporting me. NSF, Iowa State, and Alberta Livestock actually supported in part the work for Salmonella detection, and the USDA STEC-CAP grants supported work for STEC detection. So with that, I will end with my favorite GIF, which I still maintain that it accurately describes assay development, and take your questions.
Thanks, Loreen. It's great. And I love that that image at the end, and can relate very much. And anybody in this business definitely does. So I want to open this up for questions, folks. If you want to, oh, Jen, I see your hand up already. And
oh, sorry, I actually don't know how to raise my hand electronically. Sorry about that.
Just chime in,
it
folks.
Works.
Oh, yeah.
So I'm always intrigued, really nice talk, really very beautiful work. I'm always intrigued by this LPS makes holes business. And I think I heard you say that natural LPS, when it partitions from your sample that you're going to detect on the waveguide, actually doesn't make holes. But I'm kind of curious if you can turn the assay a different way. Like if you pre-make holes with LPS at a high concentration, where does the new LPS add in to that bilayer? Does it add to these holes that LPS have created, and you can kind of use that to make another detection scheme?
So are you saying, will they get bigger? Basically, if you don't backfill with lipid?
Yeah, do they get bigger? Do they even not get bigger, but rather the new LPS that you're trying to detect from your sample partitions into the edges of those holes or something?
Yeah, so this is something I'm actually working on with Kirk. And we are using a very multi-pronged approach to try to actually identify this mechanism. But essentially, LPS is always going to interact at that interface. And what we hypothesize, and is fairly well supported in the literature, is that there's some kind of nanoscale defect that's occurring right at the surface when LPS is interacting there. So if you were to say not backfill with lipid, I don't think I've ever done this, but I would definitely assume that the holes would just continue to get bigger. And that's because one of the things that happens when you incubate LPS with these lipids is that there's a lot of free lipid vesicles originally in your preparation. Even if you wash it really, really well, there's always free lipid vesicles. But LPS has what I like to call a vacuum cleaner effect on those. It will basically suck out all your free lipid vesicles and you will be left with a very clean, holy lipid bilayer. So if there's no like free vesicles, I would definitely hypothesize that the holes would just continue to grow in size because LPS would gravitate to that interface of entropy difference, free energy difference.
Is that good, Jen, or do you have a follow-up? I can see you thinking. I wasn't sure if you had a follow-up on.
I think that's an advantage for detection because then you have, a high concentration of that new LPS. And I think that that's an advantage. And I was thinking also about your microspheres with LPS holes, and then using this new idea of actually using those for sensors,
right,
to detect free solution. I think that's really cool. And I think you obviously are thinking about how to make that into a sensor.
So patterning definitely gives you an advantage because if you know where the LPS is going to go, not only can you improve the speed of that LPS association, but you can also multiplex around it. And we're actually using like gridded cover slips now to track where things are and where we expect them to show up. So,
I'll ask a question while we're seeing if anybody else chimes in here. On your electrodes, great work, Loreen. This is awesome stuff. On the electrospray or the electrodes, I guess not the sprayed ones, but the other deposition, the chemical deposition of those. When you're moving from these pyramidal to the more spongy type structures, do you have a sense? So is that the reason? Is it a lack of or a decrease in surface area that's resulting in that lower resolution that you're seeing in there?
So it's actually the printing process.
Or is it the printing process?
It's the printing process that results in the lower resolution because it's a direct inkjet printing process is what we started with. With the maskless lithography, it's a reverse pattern process, right? So instead of directly printing graphene ink, we print down this polymer and we can remove that pattern in an acetone bath. So yeah, the reason you can't directly inkjet print graphene inks in high resolution is because number one, you have to print a significant amount of layers, and two, they have this wicking coffee ring effect and the satellite droplets. So what creates the pyramid versus the pyramid structures versus the spongy structures is actually the laser annealing process. Sorry, I should go back one more. So we're using for the original process an ND1064 in the third harmonic. Don't quote me on that, pretty sure. But basically, and we look at these varying laser energy densities. And when you really turn up the energy of the laser is when you start to get these graphene petals that are pyramid structures in shape. And that's also like how you tune the superhydrophobicity of the surface. But because our IDE electrodes are really fragile, our fingers are 75 microns wide, it really just wouldn't stand up to that laser annealing process. So we had to basically take a laser that is a lot less powerful to anneal these.
Okay, and then therein, when you go to your results on that, I think they're right after there. Here. Is that where you're losing some of the quality in terms of what you're seeing? Is it the from the aerosol printed to the IML printed graphene?
Sure. So what you interpret as quality, yes. So platinum and aerosol printed graphene electrodes are extremely flat. I mean, they're almost perfectly flat. But our printed graphene electrodes are not. They have that high surface area. So that's kind of what I was indicating is that we have these semicircles here that do not appear beautiful and perfect. But the reason for that is because we have this high surface area on our electrodes.
Gotcha. Because I wanted to ask you about that too, because I'm just using, because you have high quality and good quality, but your detection limits there, I mean, they look pretty good, right? It's not,
Yeah.
yeah. So I just want to make sure I was interpreting that right. Even though they're that way, that's just due to that surface area, but they're actually really, really pretty solid in terms of that detection.
Yeah, if you know of anyone else that can do this in 15 minutes, I'd certainly like to be made aware.
Cool, very good.
Good.
Yeah, Trevor.
Loreen, fabulous talk, a great portfolio of work. Thanks for sharing it today. I had a sort of question stepping back a little bit about the sort of long-term robustness and durability of these lipid membrane sensors. We had a program with DARPA and they wanted us to make freestanding lipid membranes that could survive in the Middle East desert at 150 degrees Fahrenheit or something.
Of course.
We worked with some very clever chemists who were able to stabilize them for weeks. But in general, what's the robustness of these things for real applications?
Is not something I'm very qualified to talk about because I don't work on that project. I could call her out here because Christine worked on that project for a little bit.
I did, and it lasted only a couple days when we were working with the lipids.
So I haven't really checked in with Aaron to see about the progress of that project. But I think he was running something
a week, I believe.
So yeah, but yeah.
Okay, so there are opportunities to improve the lifetime of these things.
Of course, yeah. DARPA's not wrong. I mean, they'll ask you for the universe and beyond, but
yes, of course.
Amphiphilic polymers are where it's at, Trevor. We can talk about
you. I know. You're preaching to the choir. So
yeah, forget the lipids. Any other questions, folks?
I've been waving, so apparently I can't raise my hand because I'm a co-host. I don't have the option to raise.
Yeah, you can't raise
Zoom hand. So I was waving my real hand, but I don't think anyone saw. I have a great talk, Loreen. It was awesome. I have a little bit. So the slide you showed for the consequences of the resistance to antibiotics that's going away was of course scary beyond belief. Based on your own research, what do you think are the odds that we'll figure something out, that the numbers are not as bad as you predicted? As a mother, make me feel better about what you showed me.
Sure. Well, I was just working on this paper today. And so there's actually some interesting research that's coming out about different natural compounds that are produced by other bacteria that we're working to exploit. So there is some hope, at least on the horizon, for Pseudomonas, which is actually a really big problem. And of course, I mean, lots of money is being thrown at this. But the issue really is how quickly bacteria can develop resistance to new drugs and share that resistance with other species. And so that's one of the reasons we went after this approach, is because the alpha particles can basically release such a high energy dose inside or outside of the bacteria. We don't anticipate that they should actually be able to have time to develop any resistance to this. So hopefully, and actually, suddenly like drawing a blank as to name the pharmaceutical company that's really working on thorium for cancer therapies, but they were also interested in this for bacteria. So I don't think it's completely hopeless, but and also estimates that the world population by 2050 is actually going to meet our current predictions is falling short. So, so population decrease may actually help with that. So because we're not like sharing infections as readily.
The other good thing is that, like, I learned this from Marvel, that you could potentially turn into the Hulk or Spider-Man if it works out right. So there's that.
Wait, did you see the video of my talk?
No, but now I think I know where it's going. Hey, Alex, we have a couple of questions I want to get to. Alex and then Maria.
Yeah, my question is kind of about using the alpha particles to fight the bacteria. Kind of two sides of this question. The first one, how does it affect the other biological stuff around the bacteria? And then secondly, with that graph, I wasn't sure, does it kill all the bacteria or does it just kind of slow the growth rate compared to?
Okay, so question one, these are designed to be delivered in a very localized manner. And because we're using siderophores that are specific to bacteria, they should be taken up by bacteria as opposed to being taken up by your human tissue. Additionally, because it's an alpha emitter, it actually has a very short radioactive half-life. So in this case, it's about 18 days. And the doses that we are using are extremely small in comparison to other radioactive therapies. So anything that's left over is probably not actually going to have a large effect. And that's because alpha particles or alpha radiation is actually not effective over a large distance. So we're looking at an effective distance of somewhere between 2 and 4 microns probably. So if we can get it to where it's supposed to go, it's going to be effective against the bacteria. And you're basically, sorry, I can't remember what that's called when you kill tissue unintended. But basically there's a radius of tissue that's going to be affected around that. So we would think that that would be minimized. We actually have projects that are just now starting up to really study the effects of these in human tissue. And part two, basically what we're seeing is a bacteriostatic effect. So we haven't done any kill curves yet. And that's because we're actually just limited by how much radioactivity we can have in a building at any given time. And so we started out with these growth curves, which is pretty standard analysis. And what we see is basically we're inhibiting growth up to a certain point with various doses of the thorium-227 and different siderophores. And that's what bacteriostatic is, basically like preventing it from growing for a given amount of time. And that actually, like if you can prevent that, you can apply this kind of as an adjuvant therapy and in conjunction with other antibiotics to improve their efficacy. Does that answer your question?
Yeah, it does. Thank you.
Maria, last question.
Yes, thank you so much. My question was about the foodborne pathogen assays. Do you foresee them being used in more of the clinical setting or like the food production packing plants to, you know, help mitigate the number of lettuce recalls we see?
Yeah. So, I'm sorry, I think this is like my first spare slide. Yeah. So everybody always asks me about lettuce. Basically, these are designed to be used in a food production scenario. We haven't tested anything in clinical samples. The differentiation between a clinical sample and a food sample is like a canyon. There's a lot of different things that you have to apply to detection in human samples as opposed to just detection in food. There's standards that apply to different, the external validation companies are different. So for our purposes, we went after food because, well, number one, we got funded to go after salmonella in food. And when it comes to lettuce, I just have to address this. I actually just wrote a proposal looking at spinach, which is a little easier to manage than lettuce. So basically the problem with foodborne pathogens is that, right, the good and the bad bacteria look exactly the same. And I think that a lot of people have this fantasy that we should just be able to detect bad bacteria because they light up or something, but in reality, they all look the same. Pathogens are also very difficult to find. So, especially on an infinite surface like lettuce. So will we be able to mitigate lettuce recalls? I cannot answer that effectively, but I can tell you that billions of dollars are being thrown at that exact problem. So I'm sure we'll come to a solution one day. Right now, the way that that is done is lettuce and produce in general is actually washed, and we evaluate how many bacteria are actually in the water that's left over from that wash. So.
Awesome, thank you.
Yeah, thanks, Maria. And you know, it's a really good place then, because one thing I'm going to do, and Loreen, I'll just up your amount of work, is because for their students here, I really recommend reaching out to Loreen, because she's one of the best at really being able to identify the drivers of research. And this is something that's really important as you're learning and going through your process is what is driving research. And so she's a good person to pick her brain on this. she really knows how to think about this, including like you were showing this idea of now going to this fluorescence Deriphor type of tag. That was pretty slick. I'm going to talk to you about that after, but we don't have enough time here. Anyway, thanks, Loreen. Thanks so much, everybody. Let's thank our speaker one more time, and
Thank you.
really appreciate it. And so we will see you all next week. And Loreen, once we get past all of this, we'll have you out to visit.
Okay, sounds great.
See you all next week.
Thank you.
Thanks, Loreen. It was nice to see you.